Bom, boa tarde. Vamos continuar com a apresentação do professor, do mini curso do professor Polperieschi. Na segunda parte, o professor Polperieschi apresentar trabalho que tem a ver com o uso de chalcones como anticâncer. Então, começando agora, vamos passar a palavra para o professor Paul. Es que está apareciendo lá em cima. Aqui. What is that?
The camera is on. It is okay. It's okay. Yeah, thank you. Welcome to everybody. I continue the lectures with the second part of the series, which shows some examples, some representative cases of anti-cancer effects of natural and synthetic chalcones. First of all, let's define a little bit more precisely what cancer is at molecular biology level. All cancers, to the best of our knowledge today, derive from a single cell that have acquired the characteristics of continually dividing an unstrained manner and invading the surrounding tissues. These are very characteristic feature of cancerous cells. They behave in this abnormal manner because of the changes in the DNA sequence or structure of some key genes, which are known as cancer genes. Therefore, all cancers are genetic diseases. To understand the chemopreventive action of the so-called anti-cancer agents, let's see what do we know about the life cycle of somatic cells? The somatic cells spend their most of the time in a so-called interface, which is the over which the cell is increasing and continues its normal biochemical pathways and preparing for the division. The division of the cells is an ordered sequence of events that occur in cell preparation for division. The cell cycle is four-step process in which in the cell increasing in size, that's the so-called gap 1 or G1 state, copies DNA. This is the synthesis stage. Prepare to divide. This is the gap 2 or G2 stage, and divides, which is the mitosis stage. So these are the four sequential events, and these four defined sequential events at the same time represent the medicinal chemistry targets to modify the cell division. In the cell, when this cell division takes place, at the border of each stage, cyclin-dependent kinases collaborating with cyclins to ensure the proper timing and sequence of the cell cycle events. The cyclin-dependent kinases control the cell cycle by phosphorylating serine and threonine residues of selected proteins during different phases of the cell cycle, other proteins. You can see sometimes phosphorylation can activate, and sometimes phosphorylation inactivate these proteins. The cancer treatment strategies nowadays is fairly wide, and within each possible path, there are several different methods, different therapeutical approaches, different medicines which can used to modify the previously mentioned cell cycle of the cancerous cells. The most known therapeutical strategies involve surgery and radiation chemistry, radiation therapy, followed by chemotherapy, immunotherapy, targeted therapy, hormone therapy, and in some special cases, stem cell transplantation and precision therapy. In this lecture, in relation to chalcones, we're concentrating on the chemotherapeutical possibilities. The chemotherapeutical agents can be classified according to the structure of the chemical structure of the agent and the type of effects of them. As for the type of effects of them, there are cell cycle specific chemotherapeutical agents and non-cell cycle specific therapeutic agents. The alkylating agents, the oldest type of chemotherapeutics used in cancer, are those electrophilic derivatives which can chemically modify the proteins, the DNA, and that's the basis of the molecular mechanism of the action of these compounds. The selectivity of alkylating agents and most of the therapeutic agents as well is based on the rate of cell division of the normal cells and the cancerous cells. The rate of the cell division of the cancerous cells is higher than that of the normal cells, and that increased the rate, division rate, as provides the selectivity of this chemotherapeutic agent. The second class of these therapeutic agents represented by plant alkaloids, among them pink alkaloids, taxanes, and camptothecane alkaloids are most known and clinically used ones, which belongs to the cell cycle specific derivatives. The next one are different antibiotics like anthracyclines, chromomycins, bleomycin, mitomycin, which are also cell cycle specific, and their action is developing by a variety of different mechanisms, of different chemical mechanisms. As for flavonoids, among them, most of the more well-known anti-cancer flavonoid is quercetin, has a cell-specific effect, and this particular flavonoid, as you can see in this graph, is S-specific flavonoid. If you compare the distribution of the cells in the control cells and the quercetin treated cells, you can see that while the total amount of the cell, roughly about 14% of the total amount of the cells can be found in the S phase. However, the quercetin treated cells under very much the same conditions shows a very high increase in the S phase. This pattern is very much the same as we can observe with other flavonoids and flavonoid-related compounds like curcuminoids, resveratrol, curcumin. These are the so-called S-phase specific compounds. We go further, we can see the antimetabolites, which are also cell-specific derivatives, which are also S-phase specific derivatives, because they are intervene of the synthesis of the new DNA. The topoisomerase inhibitors, we will see them, also several flavonoids, chalcones, can possess topoisomerase inhibitor activity, as also cell cycle specific derivatives, which are intervene in the configurational and structural change of the DNA through the duplication of the DNA structure. And There is several other cell cycle specific compounds. Among them, in this lecture, I will talk about the antimicrotubule agents, which are among which several chalcones can be found. Let's see the second class of flavonoids: apigenin, acacetine, and chrysine, just to mention a few examples. As we can see, these are cyclic analogs of cyclic derivatives of chalcones. And if we have a look at these two sets of experiments, we can see that as a function of time of treatment, you see the S phase cells, amount of the cells S phase is continuously increasing after treatment of these flavonoids in both investigated cell lines. The G2M arrest is another checkpoint, okay? And this G2M arrest will be of the main spot of the cell cycle, at which not only these flavonoids, however, most of the chalcone derivatives also act. When we are talking about the anti-cancer effects of chalcones, we can distinguish the cytotoxic or anti-proliferative effect of the compounds and the chemopreventive effects of the compounds. In the present lecture, only the cytotoxic effects of the chalcones will be discussed, okay? And of which most common mechanisms are inhibition of mitosis, which manifested by means of interaction of the compounds with tubulin, uncoupling mitochondrial respiration, and induction of apoptosis, as well inhibition of DNA and RNA and protein synthesis. As for the chemopreventive effects, which are listed on the right-hand side column, a separate lecture of this series will be discussed. Let's see what are the molecular strategies, the most common molecular strategies of using chalcones as anti-cancer agents. If you look at the first picture showing the three different approaches, we can see that the first approach to develop anti-cancer chalcones is selection of natural chalcones with anti-cancer effects. Based on the most effective structures, synthetic chalcone derivatives can be and was synthesized. Modifying the chalcone structure are typically represent modification of the substitution patterns, and as it mentioned before, synthesis of cyclic rigid analogs of the compounds, or modification of the enone moiety to introduce so-called bioisoster structures. And the third approach are the so-called chalcone hybrids, in which the chalcone moiety is incorporated into another or combined with another compound who's also possess advantageous biological effects towards cancer cells. The bioisoster structures, see, and those ones which whose are equivalent isosteric in respect their biological actions. For example, here is one example of bioisosterism of chalcone, in which the enone carbon-carbon double bond was replaced by a thiazole ring, and as if you compare these IC50 values, you can see that such a modification of the chalcone structure with this bioisoster heterocyclic ring resulted the same, in most cases, much better selective cytotoxicity towards the investigated cancer cells. As for the chalcone hydrides, hybridization of the chalcone structure with other hybrid, with other molecules can be managed through covalent binding of the other structure to any of the aromatic rings, or introduction of a new structure instead of the aryl, the anon molecular part. Referring to our own results, we have investigated several open chain anti-cancer effects of several open chain chalcones and cyclic chalcones, which were already mentioned in the previous lecture. The open chain chalcones and the cyclic chalcones, the approach of this strategy was based on the previous knowledge, namely that the physicochemical properties, the reactivity of the enone unit, and the three-dimensional structure of the molecules can be modified by introducing a ring, a cyclic unit, into the open chain structure. In our experiments, the ring size were varied from 5-membered ring up to 7-membered ring, with different ring size, with different flexibility of the ring. In our original investigations, we investigated roughly about 100 derivatives of the three series. Plus, we also investigated one analogs of these chalcones, of these cyclic chalcones, in which the A ring was omitted. So only the cycloalkanone ring and the benzylidene moiety formed the investigated compounds. These are the fourth series, and in which the cycloalkanone ring, the size of the cycloalkanone ring was a 7-membered ring. This table shows the average IC50 data of the investigated compounds in this series. Here are 2 murine and 2 human lymphatic cancer cell lines. And these data represent the average of the compounds belonging to the series 1, series 3, series 2, series 3, series 4, with 5-membered ring, 6-membered ring, and 7-membered ring, with different substituents on the benzylidene unit. If we compare these average cytotoxicities, we could see that the lowest values are generally associated with the 5-membered ring and the 7-membered ring derivatives. If we compare the data of the 7-membered ring chalcone derivatives and the cycloheptanone derivatives without the A ring, we can see that removal or omission of the second aromatic ring resulted in much lower cytotoxicity towards all the 4 cell lines investigated. It gave us the proof that for successful development, we need the full chalcone moiety. In other words, we need both aromatic rings for reaching the desired cancer cell cytotoxicity. Among the 7-membered ring chalcone analogs, so-called benzosuberone derivatives, here I can show some selected the IC50 data of some selective derivatives with different substituents on the benzylidene units. If we can see these figures, we can see that the lowest IC50 derivatives, in other words, the most effective cancer cell cytotoxic effect, is associated with the methoxy and the dimethylamino derivatives. In both cases, the aromatic substituents possess lone pairs of electrons, which seems to play important part in development of the observed biological effect. Based on this previous data, we selected the 4 methoxy derivatives for further analysis. At the beginning, we performed analysis of cell cycle effect of the compounds using Jurkat T cells. The Jurkat T cells were treated with the selective benzosuberone derivative with methoxy substitution, and the cells were treated with the IC80 dose of the compounds. And we analyzed the distribution of the cells between the different phases, cell cycle phases, as shown before. What we observed that at the 24-hour samples, the cells, most of the cells could be found at the G2M phase, which is similar characteristics of those flavonoids shown before. After the 40-hour treatment, the G2M phase cells practically disappeared, and sub-G, G1 cells, and polyplural cells were found, which showed that the separation of the two daughter cells were not perfect. That why the DNA content are higher than that's in the normal cells, or the cells underwent apoptosis, which resulted in a lower DNA content. So we just cleared, we got experimental proof that our cyclic chalcone analog, the mechanism of action of the cyclic chalcone analog is very much the same as that of those flavonoids investigated before. It's known from the literature that those anti-cancer agents that stop the cell cycle at the G2M stage acts on the microtubule synthesis or microtubule degradation. The microtubules, are polymers which are developing and playing very important part in separation of the DNA content into the two daughter cells. If such separation is not perfect, the cell cycle stops, and the cell, as we could see, can undergo apoptosis. These tubulin binding drugs directly binds to tubules or inhibitors or tubulin associated kinases or inhibitors of the associated mitotic motor proteins, which results in disruption of the microtubule dynamics. They are broadly classified as microtubule stabilizing and microtubule destabilizing agents according to the effects on their tubulin polymerization and depolymerization. This figure shows the simplified structure of the microtubule, which are composed of two units, the alpha and the beta tubulin units, and the enlarged picture shows the different binding sites of microtubule stabilizers and microtubule destabilizers agent. If you can see, in this picture, we can see that taxane and apotilone are the so-called microtubule stabilizer agents, and the microtubule destabilizing agent. Among the microtubule destabilizing agents, you can find the mostly known vinca alkaloids and the colchicine. And the colchicine binding site is important from that aspect that the chalcones and the known tubulin active flavonoids binds to this colchicine binding site of the microtubules. Colchicine, as you can see, is a compound which resembles the shape and the structure of several natural products. For example, methoxyestradiol, comprestatine A4, and chalcones. All those compounds mentioned have got antitubulin activity, and binds to the colchicine binding site of the polymer or the dimer. Here is a picture which shows the binding of chalcones to the tubulin polymer, and on the left-hand side you can see some selected chalcone derivatives, which shows high binding capacity towards tubulin. What we can see, and taking into consideration of our compound, that the chalcones are substituted in the alpha position, or chalcones are also alpha substituted derivatives. However, they are cyclic ones, and they have lone substituents with lone pairs on the benzylidene unit. So in this respect, it's or structure or selective derivatives are also shows very much the same structural characteristics than those already known from the literature. Here is a picture which shows the 7 pharmacophoric points of binding to colchicine binding site. Among them, you can see the 3 hydrogen bound acceptor sites, A1, A2, and AT. The 1 hydrogen bound donor sites, this one, and 2 hydrophobic centers, H1 and H2, plus 1 planar group. So this is the molecular hint which provides the non-covalent interaction of the compounds to the colchicine binding site of the effective derivatives. Further, we investigated for compounds, the methoxy substituted benzosuberon derivatives, how the compound affects in vivo the expression of some particular genes, some particular so-called cancer genes. The genes selected were the p53, the HA-ROS, and the c-MYC genes. And here this graph shows how the expression of these genes were changed if we treated the experimental animals, mice, with control oil, with dimethyl with the polycyclic aromatic hydrocarbon dimethyl benzanthracene, DMBA, alone, and we treated the mice with dimethyl benzanthracene plus the benzylidene benzosuberon, we call it MBA, at the same and after the DMBA treatment and at the same time of the DMBA treatment and prior to the DMBA treatment. What we could see that when we treated the experimental animals with the carcinogenic dimethyl benzanthracene, the expression of these cancer genes increased according to our expectations. When, however, the experimental animals treated with these benzosuberone derivatives, these cyclic chalcone derivatives, such an increase was decreased or totally disappeared. Accordingly, our compound reduced the expression of the oncogenes that caused by the DNA damage of the dimethylbenzaltracene. That's a new feature of our compounds, and we try to understand what could be the molecular hint, the molecular mechanisms of inhibition of these inhibition of expression of these oncogenes by our cyclic chalcone derivative. Dimethylbenzaltracene is a polycyclic aromatic hydrocarbon, which is not genotoxic per se. However, it's activated by the cytochrome P451A enzymes, which oxidize the ring A, forming the 8,9 dihydrodiol as a primary product, and the epoxy derivatives of the 8,9 dihydrodiol as an ultimate carcinogen. In other words, the genotoxic effect of dimethylbenzaltracene can only be manifested when this ultimate carcinogen is formed. So, based on our previous experiments, we presumed that our methoxybenzylidene benzosuberone derivative or cyclic chalcone derivative not only affects the cell cycle, however, prevents the genotoxic, the genetic damage of the aromatic polycyclic aromatic hydrocarbons. To prove this idea, these presumptions, we investigated the cytochrome P451A inhibitory effect of the open chain methoxychalcone, the 5-membered ring methoxychalcone, the 6-membered ring, and the 7-membered ring methoxychalcones, and the well-known inhibitor of the cytochrome P450 enzyme 7,8-benzoflavone. Or presumption has been proved. As we can see, compare the Ki value of these methoxy derivatives, you can see that most of them, all of them, are even stronger inhibitor of the cytochrome P451A than the investigated 7-membered analogs. So, this data showed the cytochrome P450 compounds to become a very effective chemopreventive agents. Summarizing the result of anti-cancer effects of our cyclic chalcone analogs, we can conclude that the methoxy-substituted cyclic chalcone analogs, in particular the 7-membered analog, can affect the cell cycle of cancerous cells and stop their division at the G2M phase and prevent DNA damage of polycyclic aromatic hydrocarbons, one of the most effective and most common genotoxic carcinogen. In other words, organotoxic chalcones possess both chemopreventive and anti-cancer effects. Thank you very much for your attention.
Yo creo que no me están oyendo porque está aquí mucho. O mío está aqui muito e não consigo. Sim? Acho que sim. Bom, por hoje terminou o minicurso. Continuamos amanhã às 2h e alguns minutos para a terceira e quarta parte do minicurso. Teremos amanhã duas partes de 45 minutos, separadas por um intervalo entre 10 e 15 minutos. Alguma pergunta? No, there are not questions. No question. Bom, hasta mañana. Estoy hablando en español. Until tomorrow. In Maya is until tomorrow in Maya, who is in Maya, in Hungary. Until tomorrow. Also. No, in Maya.
In your language. In Hungarian?
Yes.
Tumor ellenes.
Tumor ellenes.
Okay. In húngaro, el profesor se despidió de todos nós. Bom, até amanhã, pessoal, e boa tarde para todos.